Ladies and gentlemen, Tim O'Brien.
Okay, thanks very much, Ger. I'm not so sure I recognize who he was introducing there, but I am delighted to be here. I've never, he said I present in other places, and I do, but I've never actually spoken in a forum like this, quite like this. So it was interesting preparing for it and putting some thoughts together as to how regenerative medicine might actually be an example of affordable innovation. So before I start, I think I might make some definitions so that we're all clear on what we're talking about. Innovation is the introduction of something new or something different. And in the context of clinical practice, you would hope to assume that that new or different activity is also useful and beneficial and represents an advance in the practice. Who should innovate? Well, really, everybody working in the health service should innovate. And that's based on the premise that right now, a lot of our clinical practice doesn't produce optimal outcomes. And if you remain static, then that will continue to be the case. Whereas if you innovate, then you can improve the lot of patients. The difficult word, it's actually off the title at the moment, but in this challenge which I chose to discuss and give some thought to, the word affordable was one of the most difficult parts. Maybe innovation is hard, but making it affordable, and let's think of affordable as actually reducing the cost of healthcare, because when we think of new technologies, we think of things that generally cost money. But could regenerative medicine be an innovation which actually reduces the cost of healthcare? So before I look to the future, because innovation and thinking about innovation is really thinking about the future. What is the future practice going to be like? I'm actually going to step back first. So I'm going to go to the past. And I'm going to ask, as I did to myself, what kind of innovations have I seen over my life? And that's kind of a depressing thought. You know, it means that I'm actually getting older, that I can now look back in life, the parts that I can remember at least. And when I look at myself in the mirror, I'm absolutely convinced I look the exact same as all of you, but a lot of people would disagree. And so there have been years that have passed by. And you know, the key innovation for me that really stands out, and I'm sure it's evident to most of the older people in the audience as well, maybe less so to you guys and girls, who are somewhat younger, which is connectivity, communications. It's hard. I left, like Larry, I moved to the Mayo Clinic in 1988, which was before Larry. Larry was making his confirmation that year, but I moved to the Mayo Clinic, and there were no PCs, there were no desktop PCs, there were no what are the things you carry around with you? The laptops. There were none of those. There was no mobile phones. There was no email. I mean, that's 1988. That's not, you know, it's not a lifetime ago but that's, and most of you have grown up with all those things. So I think that's a disruptive technology, a transformative technology, an innovation of the highest level, essentially. And then if I think about medical practice, you know, and this is interesting to do when you're as old as I am, you can look back and say, well, what did we do back then that is kind of different now because of the innovations that have occurred? And there are many examples, and all of you, I'm sure, would throw out maybe different ones, but I'm going to give maybe 2 or 3 examples of how the practice of medicine now is so different to when I was an SHO, and that's only, what is it, 20 plus years ago. So let me give you some clinical examples. One is in Larry's area of gastroenterology, and every single night when I was a surgical intern or when I was a medical SHO, bleeding peptic ulcer was one of the most common admissions. I can't remember the last time I've seen one now, and that arose from the Nobel Prize winning observation by Marshall and Robin in Australia, that these little curved rods in the stomach, which seem to be associated with inflammation, might be linked. I mean, if you said to us when we were interns, SHO medical students, peptic ulcer disease is an infectious disease, you'd be laughed at. And the pathologist was Robin. He made the observation under the microscope, and Marshall was just a young clinical trainee who was kind of interested by this, and they went and pursued it and proved that Helicobacter was the cause of peptic ulcer disease. Now we treat it with antibiotics, and we don't have the bleeding peptic ulcers coming in as often, and the surgical trainees don't need to spend their time learning about highly selective vagotomies and pyloroplasty and all those things that were part and parcel of what peptic ulcer disease was then. I could have predicted when I was an SHO that that's the way things would have gone. Glucose monitoring, some of you will know that I have a minor passing interest in diabetes, but glucose monitoring, we admitted patients to hospital for 2 days to get glucose monitoring. So I was in an SHO in the diabetes clinic. Patient comes in, has very bad control. They didn't have home monitoring at that point. I'm really making myself sound like a dinosaur with no computers and no home monitoring, but we admitted them to hospital. Their glucose levels were fantastic for the 2 days they were in the hospital, surprisingly, when they were exposed to the culinary delights of Cork University Hospital. And then they went off home again. And then a third example in endocrine practice of the way practice has changed would be hyperparathyroidism. So when I was in SHO, we were seeing patients with pepper pot skulls and bony erosions and renal impairment and bone stones, Mohs abdominal groans is what they came with. We didn't have a PTH assay, so it was quite difficult to differentiate between cancer and hyperparathyroidism. So 3 examples of innovations which have essentially radically changed the way we practice medicine. And you're so young you can't really look back at your life yet and see what has changed. But it's interesting for you to look forward and wonder, what is it that's going to be different 20 years from now? And so if I could step away from the past and kind of move into the future again, and what are the challenges that face us as a society? The major challenge is the burgeoning cost of healthcare. So as Chair mentioned, I just came back from New York. I was at a meeting where I was listening to an investment banker. What kind of a meeting was that? But I was listening to an investment banker, and the investment banker was pointing out in the US they spend $3 trillion a year at the moment on healthcare, 17.4% of their GDP. That's going to predicted to be $6 trillion in 2030. So that's staggering amount of money. And the main reason for it is not new technologies, actually. The main reason for it is the aging population, demographics. You're going to double the number of elderly people in the US over that period of time, and they're all going to have chronic diseases, the diseases that Larry alluded to, chronic degenerative diseases. And the kind of pattern up to now for the management of chronic disease has resulted in what might be viewed as incremental innovation, which aimed at trying to slow the progression of disease or avoid the complications of disease, but didn't deal with the underlying disease itself. And that's where the paradigm shift, which I would suggest that regenerative medicine represents, that's where the paradigm shift is going to occur, and where it could actually represent affordable innovation. It's not just you just deliver another layer of cost to the system, but that you fundamentally alter and affect the underlying disease. So this is the meeting I was at. It was the Alliance for Regenerative Medicine that I was at for the last 2 days. And so I borrowed, plagiarized, it can't be plagiarism if I'm admitting I did it, from their annual report, a definition of regenerative medicine. So it aims to augment, repair, replace, or regenerate organs and tissues that have been damaged by disease, injury, or natural aging. And it represents a new paradigm in human health with the potential to resolve unmet medical needs by addressing the underlying cause of disease. Let me just point out again, just to give examples of what are unmet medical needs that we deal with in our clinical practice every day at the moment. I can tell you about a patient of mine who has 35 years of type 1 diabetes. He's driving down the road with his wife in the passenger seat, and he starts to drive erratically, going faster and faster, and he's sweating, and he's getting verbally abusive to her. And she knows he's having a hypo, but he doesn't know he's having a hypo. As she tries to convince him to go off the road, he just drives faster. And that's not just because he's a husband, because husbands kind of do these things. This is because he was having a hypo, just for the medical students who mightn't have attended my hypoglycemia lectures. But that patient is on state-of-the-art, innovative, multiple daily injection program, fully educated, and that to me represents an unmet medical need. I can give you a second scenario which relates to a colleague of Matt and Larry and myself at the Mayo Clinic who actually presented with a condition where he noticed that he was kneeling down at Mass and he couldn't get up one day because he was weak, and he had been fasciculating. He had these little fasciculations, and within 2 years he was dead from motor neuron disease while waiting to try to get into a clinical trial in regenerative medicine. And then the third example I'll allude to in terms of unmet medical need is critical limb ischemia. So every 20 seconds, somebody somewhere in the world loses a leg. Not loses it, it's not like, where did I put my leg? But actually had an amputation. Every 20 seconds. So if you look at the length of time that I'm giving this talk, for in 1 minute there'll be 3 legs amputated. And one might wonder, is that a good treatment? I mean, is that the best we can do? You know, this audience full of, you know, bright, young, enthusiastic people, are you going to be happy that motor neuron disease, we can do nothing for it other than watch the inexorable decline towards death? You know, that my type 1 diabetic has to, and his wife and family have to cope with the difficulties having, and that we're sawing through a leg and throwing it out in 2013. That to me represents unmet medical need. And therefore, this area of regenerative medicine, which is meant to focus on the degenerative, the chronic diseases, and to impact the underlying disease process. So this is, if your practice was looking after hydractinia, and Yuri Frank is a developmental biologist here in Galway, and Yuri Frank works with hydractinia, which is a marine organism, and on the left, you see the normal. They've got a head and they've got a kind of a body. And Yuri went and decapitated him there in the second picture just for the hell of it. So just took the head off. And then 2 days later, the head is back. I mean, that to me is kind of exciting, that you remove somebody's head and 2 days later, the head is faithfully restored. And that process is driven by stem cells. So the injury results in signals that cause the stem cells to migrate up through the body and start differentiating into the specialized tissues that form the head. That's hydractinia, so I don't know that anytime soon we'll be saying you can just chop, you know, one of your heads off and 2 days later, lo and behold, you're back again. But it's an example of the kind of exciting underlying fundamental science which I'm going to now also show a video, if we can get it going. This is just downloaded from the net, and it's another example of tissue regeneration. It'll take about a minute.
We're going to have a look at the way in which an amputated limb grows over the course of about 90 days in a salamander. That's a time-lapse movie, watching that thing grow. And now we're going to see what's really going on. Here's the salamander, and it's got a completely new limb. It looks perfect. It has inside bone, it has nerves and muscle, and it can even wiggle. In fact, it's perfect. Now we're going to cut it off. But here's the good news. It grows back. So don't be too scared. First, wound healing. Did you see that wound heal? Now we're watching what the nuke can do we cannot do. Cells are streaming out of the surrounding tissues into the area of the wound and forming what we call a blasphema, which is a group of undifferentiated cells that are in fact really just like stem cells. And they're multicolored because they come from skin, from muscle, and even from cartilage. And these have a miraculous memory of what they used to be.
So wouldn't it be great if an amputated piece of heart or an amputated piece of brain or an amputated leg would just regenerate? And so somehow in going evolutionarily from Hydractinia to newt salamander, we lose it as we become mammalian. But yet many of the genes that are involved and many of the cellular activities that are involved still remain in a homologous sense in humans. So people like Yuri Frank and the Drosophila developmental biologists, the zebrafish, they're really describing fundamental processes which will probably teach us a lot in terms of how to reach this point someday. So another important thing in the context of the basic fundamental science underpinning regeneration is what I'm depicting on this slide as a multidisciplinary interaction. So this is an interaction with, that we have in REMEDI with Walter Kolch and Boris Kolodenko from Austria and Russia, now working in Dublin. And they're mathematicians and they're modelers, and they work with us biologists, and we can't understand what each other is saying, which is a little bit challenging. But the next generation of PhD students who come out from under this combined program will actually be bilingual. Right now we can only speak our language, and when I see a model I zone out, all those formulas and such. But what we do is we generate experimental data, and then the modelers, the mathematicians, will draft a model around that, and then we'll try validate their model, and then that allows you to go on and refine and expand. And it looks at cellular activities and physiology in a very integrated manner, not in a linear, one thing binds to another thing and then has an effect, but rather all the multitude of things that are playing into it. So developmental biology, systems biology, immunology, which Matt hails from, they're all fundamental sciences which will be necessary to bring forward regenerative medicine. The technologies that will be applied in the treatment will include cell therapy, biomaterials, medical device technology, gene therapy, biologics, and small molecules. And cell therapy is probably the largest part of it at the moment. There's already about 50 products licensed, proven, and in use right now to regenerate tissue, largely in the wound healing and bone repair areas, and all the other activity in heart repair, brain repair, beta cell repair. They're all happening in the clinical trial arena. So as cells, and cell therapy is one of the major technologies which is driving forward this regenerative medicine paradigm. And as I emphasize, the regenerative medicine paradigm is that you influence the underlying disease and restore tissue to normality. And therefore, that should be cost saving as opposed to the ongoing need for chronic treatment, which is necessary when you don't impact the underlying disease. These are embryonic stem cells, mouse embryonic stem cells in this slide, and they're derived from the early blastocyst and require the removal of the cells of the early blastocyst, which is why there's such an ethical controversy about whether it's okay to do it or not. But the embryonic stem cell is totipotent, so you have the capacity to grow whatever tissue you want and then transplant that. This is probably the most exciting technology that has hit the regenerative medicine space, which is the induction of pluripotency. And this resulted in the Nobel Prize for Physiology and Medicine last year for Shinya Yamanaka and John Gurdon. John Gurdon in 1958 had shown that you can clone frogs, and Shinya Yamanaka, kind of following up on that work, showed that you could put 4 factors into a cell and you could convert it back to totipotency. And they do this now with skin fibroblasts. And again, if you were to say to me 20 years ago, 10 years ago, Yamanaka published this in 2007, so this is just 5 years old, and he has a Nobel Prize within 5 years because of the fundamental paradigm shift that's being created by you can now take a somatic cell and convert it back to a totipotent stage from which it can then be driven forward. So every one of us can have a skin biopsy, can have a totipotent cell created out of a skin biopsy, and then heart, lung, liver, bone, kidney grown in a dish. That is phenomenal and would have been science fiction up to Yamanaka's work. It's now progressed to the point where now you don't actually have to go back to the totipotent stage. It's now known that you can put factors that will drive it to specifically the lineage you want. So you can drive this skin fibroblast to a neuron, so you don't have to go all the way back to totipotency and then forward to neuronal differentiation. So this is published in the last year or two. So this field is really developing rapidly, and suggests then that there may be an endless source for all of us to have our own tissues replaced, repaired, regenerated in this manner. There is a little caveat, and that is putting in the 4 factors requires lentiviral, retroviral vectors, which are pretty toxic at the moment. But that technology is being worked on. There are also adult stem cells, which are not totipotent. So this is the cell that we work with in REMEDI most of the time. It's a bone marrow-derived mesenchymal stem cell. So you take 20 mils of marrow, plate the cells on plastic, and the cells stick to the plastic, and the hematopoietic stem cells float away. And then you're left, and you can culture expand these to trillions over a 4 to 5 week period. So this is another example of a cell which may be used therapeutically. It doesn't have totipotency. It can become bone cartilage and adipose, the stuff that Francis was talking about earlier, which can actually be generated quite effectively by eating McDonald's. But you can also generate adipocytes by differentiating MSCs into that lineage. Now, another very exciting current paradigm shift that's occurring in the stem cell field is that stem cell therapies may not only be beneficial to regenerate tissue by differentiation, but actually these cells, all of these individual cells, secrete a whole host of factors which are pro-healing and pro-reparative, and anti-inflammatory and immunomodulatory and pro-angiogenic. So now you're looking at, again, a new concept in disease, that these cells may be used as a mini factory to produce a host of tissue elements that will aid repair and so not only might the cell differentiate down towards the bone or cartilage in the case of this cell, but it can make stuff. That doesn't sound terribly scientific, but it makes stuff, and some of the stuff is good stuff. It can induce new vessel formation. It's anti-inflammatory, immunomodulatory, and there's a multitude of clinical trials. In fact, I don't think you will find a specialty of medicine which is not being studied at the moment with this cell type, because of the paracrine secretory factors. And in fact, not only are there paracrine effects, so if you think of autocrine as cell secretes effect on the cell, paracrine cell secretes effect on local cells, endocrine, which is what Dr. Finucane and myself spend our time doing, this is kind of inducing an endocrine state by putting these cells in. Not only do the cells secrete factors which have a local beneficial effect, they actually travel to distal sites as well. So it's almost like an artificial endocrine system which has been introduced. So regenerative medicine then, I talked about the technologies would require cells or genes or biomaterials or devices and small molecules, biologics, with the underlying goal being to restore the tissue state to normal. I don't think we're going to see that happen overnight in the same way that hydractinia replaces its head, or newts and salamander will replace a limb over 90 days. The hydractinia does it in 2 days. But you will see the current 50 to 70 currently approved products with now hundreds of trials going on. You will see a gradual encroachment from the clinical trial area into clinical practice. But this is the kind of ecosystem that's required, and this is the ecosystem that we've worked on establishing in Galway. It includes advances in fundamental science, the developmental biologists, the immunologists, the neuroscientists talking to each other, a clinical trials network, so the ability to take the preclinical observations and put them into trials, a skilled workforce and a primed health service. So our education efforts are hoping to drive the future generation of regenerative medicine pioneers and entrepreneurs. I'm looking at 4 or 5 of you there at the moment. I didn't say which 4 or 5. The the industrialization of product manufacture, so the cells have to be grown. And this is one of the key challenges as a cell therapy. So if you think of the Lipitor that you prescribe for a patient, they open the container, they take the 20 milligrams of Lipitor, they swallow it. Well, that has to be it has to be made somewhere, it has to be transported. With cells, it's much more difficult because it's a living treatment, and you can't end sterilize it. So you can't make it. We've built the GMP facility in the university, and in the GMP facility we grow these cells, but you can't end sterilize them because you kill them, and then they're no longer therapeutically effective. We need innovative regulatory strategies, innovative business models and reimbursement strategies. So if we develop a fantastic regenerative approach to restoring something, and it costs so much, and there's a product called Provenge, which is a cell therapy, immunotherapy product for advanced prostate cancer, but it's very expensive, and it's not fundamentally altering the underlying course of disease. So you got to get reimbursed if this is ever going to work. That's why I was listening to an investment banker yesterday. You have to demonstrate cost saving through robust health economic analysis. So we're across the road from the Cairns building, and those people in health technology assessment, health economics are crucial to this. You can think of, if you do have a replacement beta cell, so let's say you use embryonic stem cell, drive it down to beta cells, and now you deliver that to a patient in some kind of a capsule, and you cure their type 1 diabetes, then what cost should that carry relative to the savings that will ensue, because the patient won't get retinopathy, nephropathy, neuropathy, stroke, heart attack, and will have a completely altered life. So the element of cost of product, cost of goods versus what you're doing to healthcare dollars, or euro in our case, where you see healthcare expenditure only going up, is regenerative medicine an affordable innovation that will inflect or deflect that increase in cost of healthcare? And I would suggest that it is. This is an example of a program that we have started in Galway. It's led by Galway, but has other Irish universities involved. It's called BioInnovate, and it brings together an engineer, a law graduate, a medicine graduate, and there's 4 of them anyway. There's law, business, science, medicine, or engineering. There's 4 of them together. They form a team, and they spend a year doing analysis of things that need innovative solutions, and then they try spin out a company at the end of the year. So this is where you're bringing research and clinical practice and academia together with business and entrepreneurship and innovation. And we've run it now. Faisal Sharif, who's in cardiology, is the medical director. Mark Burzy, an engineer, is the director. And they've spun out now a number of activities in just 3 years of its existence. So I'm going to conclude. Before I actually focus on what's on the slide, I do want to put in a caveat. And the caveat is, anything that sounds too good to be true is probably too good to be true. So I want to step back a little bit from my incredibly enthusiastic endorsement of all of your hearts, livers, lungs. You can expose it to any toxin you want, because you're just going to rip a bit of skin out, grow another liver, and shove it back in. So there will be some, I think the reality will be somewhere between that and us not being able to do anything right now. But the real victims sometimes in this hype are the patients who develop enormous expectations. And we have patients come to us every week now, weekly, daily emails, wanting stem cells for their loved relative who's had a stroke, or who has end-stage cirrhosis, or who's on dialysis, or who needs a hip replacement, and they want stem cells to fix it. Motor neuron disease patients, families contacting us saying, just give us the stem cells. We don't care whether it's proven or not. We know that in 2 years, this relative of ours is going to be dead. So give us the stem cells. But the translational paradigm in our Western societies is to do preclinical efficacy, preclinical toxicology in animals, phase 1, phase 2, phase 3. And I do case studies in our Masters of Regenerative Medicine with students, which from the first publication to the phase 3 study is 20 years. So I'm faced with people with motor neuron disease saying they want the therapy and they've got 2 years to live. And I said, but it's going to take 20 years so there's a real tension there between what patients want. And that's where stem cell tourism comes from. So the concept of, and you can go on websites in China, Ukraine, Belize, number of places, and you can see you deposit the 20,000 euro, and then you arrive there. They pick you up at the airport, and they bring you, and on day 1 you'll have your first stem cell infusion. You know where the stem cells are coming from, you know who they're coming from, you know what kind of quality control was necessarily, and there definitely isn't the clinical trial paradigm of preclinical efficacy, preclinical toxicology, phase 1, 2, 3. The reason that's there is to safeguard patients, but we have to work as academics in some way to try and catapult that in terms of time. So should it take as long as it takes us to do preclinical efficacy? Or should we get more efficient at doing it? And we're putting a lot of thought and effort into that. So with that caveat in mind, I do believe that affordable innovation is crucial to contain future costs of healthcare. $6 trillion, currently 17.4% of GDP, is not sustainable. And right now the solution to it, the solution being presented is spend less, ration, reduce the reimbursement costs, even to levels below what it costs to deliver. So is that a solution? Is the solution to the rising costs? We're getting older, we're living longer, we have more chronic degenerative disease. Is the solution to just say, well, we're not going to pay anymore, we're going to pay less, we're going to have restrictions, we're going to have rationing? Or is there a fundamental paradigm shift that can occur where you harness the regenerative technology, as I spoke about, stem cells, gene therapy, biomaterials, scaffolds, combinations thereof? And I suspect in 20 years' time, where I saw the difference between bleeding peptic ulcers every single night to where I don't see bleeding peptic ulcers anymore, you may well be, in your practice, delivering scaffolds with cells on them, scaffolds with genetically modified cells on them. Now that process with the regulatory hurdles will be substantial, because when you go to a regulator and say you want to put a stem cell or you want to put a gene, they kind of get scared. When you tell them you want to put a genetically modified stem cell on a scaffold through a device, they're even more scared. So there is a challenge there, but the focus will need to be the development of more effective treatments of chronic and burdensome diseases which reduce cost of healthcare rather than adding to the cost of healthcare. Thank you.